Okay, so what we have here are litter bags, and these are made out of fiberglass window screen, and they're glued on two sides with a hot glue gun. This keeps the material from decomposing when it's buried, and we'll use these to put our organic samples in. So we'll open them up, and we have three different kinds of material. We have two plant and one animal in the form of eggshells. So we have something that's very fibrous, and we have something that is more easily digested plant material, and some animal material in the form of the eggshells. So we open up the bag, and we can take our material and place it inside of these bags. So we can place in some cereal in one. Okay. We take the dried plant leaves. These are oak leaves. You're just going to take them and you're going to fold them up and place them inside of one of the individual bags as best you can. Just kind of take it and put it in there. Okay. And for the last one, we're going to put in the eggshells. So just take some of this. It's not really maybe quite enough there, so we're going to grab one more piece and stick a little bit of extra into this one bag. And then once we have these, we're going to take them over and seal them with a hot glue gun, and then we'll weigh them so we know the starting mass. So once we have these bags sealed with the hot glue gun, we take them over to the balance and determine the mass of each of these bags. So we're starting off with the one that has the cereal in it. We place it on the balance, and we'll take the starting weight. And that one is 2.3 grams, I think it was. Okay, and here we have the one with the leaf sample in it. Take that mass, and the same thing goes here with the eggshell. So now we have our starting mass, so as our base point for decomposition. And from here, these samples can be taken and then buried into our bio columns. Okay, then for environmental conditions, we are going to have 4 different environmental conditions. We have 2 temperatures and 2 different levels of water. So for temperature, we have room and refrigerator temperature. And then for water, we're going to have really wet, and then we're going to have M for moist. And so what we'll have is 1 person per table will do one of each of these treatments. So we'll have cold, room temperature, wet, and moist. Now what we're going to do is set up our column, and the first thing we'll determine is the moisture level. So if we're going to keep it saturated, we will keep a lid on this portion of the bottle so that as we put moisture in it, we can keep the water level filled so that we don't have any drainage, and this is going to stay wet. And other ones, we're going to want to allow moisture to move through. We're going to keep in the very bottom. We'll keep a reservoir of water. We'll remove this lid, and we'll have a screen that we'll place in so that the soil does not fall down through into the reservoir. Okay, so we're going to have a reservoir in the bottom that's filled with water, and then we're going to have string that's going to act as a wick. This is cotton string that will absorb the moisture. So we're taking 2 levels, just 2 pieces of string. It's actually 1 folded over. We'll make a small little cut in the screen so that we can thread the string through it. We take that and pull it through. And once this is pulled through, we stick this down through the funnel-shaped portion of the bottle where the soil is going to go. Close that up. And now we're ready to add some potting soil to this container. So you come over to the moistened potting soil. We'll take a container. I'm going to try to hold the wick up somewhat as we put it in so that the wick stays in contact with the potting soil. So I'm reaching down in here and pulling the wick up so that it stays in contact with the soil. Add a little bit more potting soil. And once that's in there, then we're ready to start taking our sample bags, and the sample bags can be buried in this mix. Okay, with the wick hanging out the bottom, take it over and we'll take these bags that we have ready and just start placing them down in the soil, and if we need more we can always add more potting soil on top, but we want to try to get good contact to all sides of the litter bags in contact with the potting soil. So just bury them all in there. And we're probably going to need to add a little bit of extra potting soil to the top. You want to try to get these as evenly spaced as possible with good contact with the soil. To cover up our litter bags and tamping it down so we have good contact with between the soil and the organic matter. And the final step is to fill our reservoir in the bottom. So we just add water to this reservoir, which shouldn't really evaporate because it's going to be sealed, and we're going to tape it with the upper part here. And our wick will go down into that moisture, and as the water evaporates from the soil or is used, it's going to be replaced by the reservoir. And finally, just to make sure that we have everything together, we're going to water the top in a little bit, very gently so we don't disturb the surface. Just kind of add a little extra moisture. Any extra is going to run down into the reservoir. And now we have a complete eco column ready to go.